Alright, this is the first video of DNA replication. In this video, we're going to cover some vocabulary that's very important to understand. But I'm going to stop this video right before transcription to only cover DNA, the basic part of replication. Alright, now when the body starts to go through the process of mitosis to copy cells, a cell's going to spend about 90% of its lifetime through mitosis, through the interphase portion of mitosis. Now it does that because then when DNA needs to replicate, that's a more lengthy process to take part, and sometimes cells are under certain situations where time might be an issue. And so therefore, replication is always occurring inside of our cells. Remember that it's DNA that is replicating, and that's basically the genetic instructions of your body. And so because of its complexity and so forth, it needs to take time, and it needs to be ready when mitosis is going to occur. Now let me quickly draw something real quick. We know a chromosome, when you look at a chromosome, as being something more or less like this. Okay? Now, in reality, what this is made of is, if you think about it, it's this string type material of all your DNA, all your chromosomes, basically bundled up almost like a big ball of yarn, if you will, but instead of a ball, it's more like a cylindrical shape. Now, there was a tip where I started here in the beginning, right? And one would think logically, if DNA is going to replicate, why not just start to copy and extend this tip outward right there? DNA doesn't do that. And there's a couple different reasons why, which we get into later. But DNA actually will copy somewhere here in the middle. And I don't mean somewhere like one location, but several parts. There are certain specific areas where DNA will start to copy. So if I take this and I kind of look at it microscopically, you kind of blow it up under the lens of a microscope, it would appear something more like this. So here you have your double helix of DNA, and basically this gets smaller and smaller, and your chromosome is that long string-like material that is really all this kind of just bundled up in here. All 46 of your chromosomes are like that. Now, through the process of mitosis, when this chromosome turns into a tetrad, and excuse the drawing, and eventually splits at the centromere here, we have to make sure that the genetic material in here is exactly like the genetic material in here. And if you elongate this and continue growing it this way, you may not have that. But if DNA replicates by taking this and just kind of bringing it out over here so that we would look at it there, and what you would have is this string out like that, well, if you split it, okay, and then have your DNA looking this way, and then we split it one more time here, a complementary side will build to this part, and a complementary side will build to this part. You're going to see that this side will eventually look exactly like this side. And that's what ends up happening when this chromosome ends up splitting up. So DNA replication doesn't take the tip and just start continuing the sequence, because you may or may not have identical sides that way. DNA splits in the middle. Now throughout this, let's keep one thing in mind, this question, which is basically discussing what I was just discussing with you. How does replication ensure that identical copies of DNA are gonna be made once replication has completed? When your DNA splits here, when that double helix splits up and forms what we call a replication fork, you see the rungs of DNA here, they split up, and a complementary side is going to be attached to it, which these fragments are going to be basically like this side. And then the same thing is going to be happening over here. After the process is done, this will wind back up, and DNA in the end will become bigger or lengthier, if you will, that way. So that therefore, once the cell needs to go through mitosis, the DNA has already replicated for it, and so therefore is ready. The copy now has more DNA to split up that way. All right. So let's go through some of these words that we need to know. First, the double helix is what we have here. It is your quintessential picture of DNA that you've seen in 1,000 books and pictures and so forth. That's your double helix. We would refer to it as kind of looking like a twisted ladder. You have your nucleotides, which are your subunits of the nucleic acid molecule. That's going to consist of your sugars, your phosphate group, and your nitrogenous bases. Okay? And then you have your nucleoside, which is your subunit of basically the same thing, the sugar, the nitrogenous base, but the nucleoside doesn't consist of the phosphate portion of DNA. Okay? Replication, you could use the same definition of replication that you have in an English class, to copy, to duplicate, and so forth. So then your replication fork, here's where that expression, the fork in the road, comes into play. This is your replication fork. You can see that this is going to continue this way, and we split this double helix into half going this way, half going that way. Kind of resembles a fork. This site where this occurs, where the split occurs, is called a replication fork. An enzyme, we go back to macromolecules, same definition. The enzyme is basically that protein that facilitates a chemical reaction by making it occur faster without changing the identity of the chemicals. And remember that enzymes also not only speed up the process, but also require what? Less activation energy in order to occur as well. When this replication fork occurs, when the split occurs, leading toward the replication fork, you're going to have a leading strand. A leading strand is a DNA strand that forms as a continuous strand in the direction toward your replication fork. On the opposite end, you have your lagging strand that builds away from the replication fork. Keep in mind that when this occurs, this fork also unites further down in the DNA. Okay? So your replication, the process of duplicating, replication fork, where the split occurs, the enzyme like helicase, for example, ligase, and so forth, we're going to get into now, are proteins that facilitate for the whole thing to occur, and you're going to have a lagging strand moving away from the fork and a leading strand that moves toward. These parts right here are what we call Okazaki fragments. These are the fragments, those complementary fragments that are going to come build and attach to this. Now they can't attach any way. There is a specific rule as to how they can attach. We'll get into in just a little bit. So again, here's your DNA is your long molecule made up of these units called these nucleotides. Your nucleotide is going to be consisting of your 5-carbon sugar, okay? Your 5-carbon sugar, your phosphate group. Sorry, I lose my cursor here. Here's your 5-carbon sugar. Here's your phosphate group, and you have your nitrogenous base. There are basically, in DNA, there are basically 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. There's a rule as to who goes with who. The quick way that you could learn this is, first of all, we only use the first letter, A, G, C, and T. If you think back to when we were doing macromolecules, if you think back to that real big wheel that had the various colors on it, and in the very center there was a very large A, T, C, and G. And remember, we learned what a codon was back then, which was what? The 3 letters of these nitrogenous bases, paired up to make the different amino acids. Well, there are rules as to how these things can pair up. So if you take these 4 letters, A, G, C, and T, which 2 letters can I combine to make a word? The only 2. If I combine A and G or G and A, I don't have a word. If I combine G and C or C and T or T and C or C and T or whatever, doesn't matter the order, I don't have a word. Except if I combine A and T, I have what? I have the word at. So those 2 are the only 2 that combine to make a word. Therefore, the easy way to remember is A can only be linked up with T. So by default, C and G can only be linked up together. You see, A only fits together complementing T, and C only goes with G. If I've got a G here, I can't have anything other than a C on this side. You cannot have a T here. You cannot have an A here. Okay? Again, I'm only discussing DNA right now. When we get into RNA, that kind of changes a little bit. But for right now, I just want to keep your mindset in DNA. So, the rule is A complements together with T and G complements together with C. It is said that DNA has a specific puzzle-like shape for this rule to apply. If you notice, and that would be true, if you notice the shape of A, out of this shape, and this shape, the only one that's going to match with A is going to be what? Is going to be T. If I brought cytosine to here, it's gonna be like this. And does that work? Does that look like it locks? No, it doesn't. And if I brought any other one, or G for example, would be, still doesn't quite lock as good as T, which is going to lock right in, right there. Okay? So, when this builds up, and you can see it here, A and T, I don't know if you're going to be able to see, but there's a line right there, which is kind of like that. And in C and G, it'll be their shapes. So they pair up to make this DNA. Now during DNA replication, they're going to produce complementary strands. I had mentioned complementary strands earlier. Right here at the replication fork, this side comes this way, and you're going to have a leading strand. There's your complementary strand right there. Basically, how it works is this. Okay? If this one, right, let's take this one right here. You see this leading strand? Okay? It's gonna be added, and when we get into transcription and translation, you'll understand how that works. Because remember, keep in mind, DNA is stored in the nucleus, but these things are built outside of the nucleus, and these strands have to come in and do that. There's special proteins that allow for that to happen. So again, we'll get there. Anyway, when this splits up, imagine the blue is not there. Or imagine the blue is going to be continued. All right, let's say this one right here is A. And this second one is a C. And this one is another A. Right here, this complementary strand, what would have to go there? What would be the one that would have to pair up on this side to these three? That would be this blue one continuing over this way and build here, here, and here. All right? So that it continues just like it did right there. Right there. This whole thing right there. But what would have to be the acids? See what would have to be the nucleic acids here? ACA. The complementary side would have to be, T, G, T. Okay? So when this one right here, TGT, which is going to be floating around somewhere else in the cell, and it's going to be brought here by a protein, it's going to attach. Remember that we work in 3 for like a codon, okay? So it's going to attach. Then the next one here, it could be something else. Again, this complementary side here must be a specific order of them to form the proper nucleotides, okay? So, each strand of DNA, basically this serves as a template. If I give you this template, then you would automatically be able to tell me what goes on this side. And the same thing over here, correct? The DNA splits, replication fork, half goes this way, half goes this way. This half serves as a template. So when you've got these sticking out this way, G, C, C, I know the complementary side is going to have to be C, G, and G. Once this split occurs, this strand basically serves as a template for the new strand. And they're going to be these enzymes that are going to come. They almost look like this. It's a really bad drawing, but. And they'll come and they'll simply attach to right there. And this attachment is what starts to build this actual leading and lagging strand. Okay? All right. So here are the big major parts that occur here. All right? DNA helicase is going to be in that last picture can I go back? This is helicase right here. So DNA helicase is going to bind to the double-stranded DNA, all right? And it's going to basically stimulate the separation of the two strands, okay? Now, that's all helicase does. Once helicase does this part, primase comes and initiates replication by adding short RNA fragments, okay? They're called primers, and it adds it to the old strands. But guess what? Primase isn't always perfect. Sometimes it can make mistakes. So then, polymerase comes and it'll join individual nucleotides to produce the new strand using the old strand as a template, and they're also going to proofread and repair. Later we'll talk about omissions, deletions, and things, substitutions, things like that. Those are the types of errors that can occur here. So polymerase is going to come and proofread what primase did. It's going to make any repairs that it needs to. Ligase will basically assume that polymerase did a good job, made everything correct, and then what ligase starts to do is it starts to finalize the process. It just starts to link them all together. Okay? It's going to bind those DNA fragments together by adding phosphate into these groups to make that backbone of that sugar for DNA. Okay? So let's go a little more one by one into these four. These four are really big. It's really important to know them. So, helicase. Well, when DNA replication begins at the places called the origins, within the DNA molecule, the creation, within the DNA molecule and the creation of the replication forks, helicase is going to do his job there. Okay? The strand separation is going to be made possible because helicase is basically the one that takes this DNA. Here's our twisted ladder. Here of DNA, right? And this is kind of the big picture of it. So when you see this in a with less magnification, you just basically see it as this, okay? Kind of like this, real small. So what helicase is going to do is just going to split it like this. And then later, those Okazaki fragments are going to come and attach this side to this side, and the same thing over here. But what helicase does is it creates this split right there. That's the biggest job helicase does. Helicase is going to create that split right there, and then primase comes and basically takes these strands and places them here. Okay? Now primase is supposed to be the one that looks and sees A, so it has to bring a T. Most of the time, it's good with that. But sometimes it can make a mistake and bring the wrong one. What happened? Oh, okay, whatever that was. Anyway, so there will be an error here. Again, all primase does is just basically bring these primers up to this point right here, and that's it. Now, these primers are synthesized by primase. Okay, so they're going to be made by primase, initiating the replication process. Okay, after that, primase is basically done. It has completed its part, and then polymerase comes and is basically going to proofread and repair everything that primase does. Primase is going to basically align. When I say proofread and repair, what could have happened was if you've got this strand, I'll just use this picture here to save time. Now this thing won't disappear. Alright, if you've got this lined up right there, what primase may have put the strand over here, but instead of being these three, it's supposed to be these three. Well, polymerase will take it and move it and put it to the location where it's supposed to be. So polymerase is what's considered the most important enzyme, because this is the one that catches the mistakes that primase does. Okay? It's going to basically line them up. Remember, these are not attached, so we say they're unzipped at this point. That's ligase's job. They're unzipped at this point, and it's going to basically It has the job of adding whatever needs to be added, reading the strands, making sure there are no mistakes, realigning, repairing, proofreading, make sure everything is good. I would venture to say polymerase is probably the most important enzyme there is. Okay. Afterward, ligase basically comes and connects. This is the one that does zip that complementary leading and lagging strand to the older strand of DNA. It binds it and adds phosphate in the little gaps to make that phosphate ribose sugar backbone of DNA. It basically complements, or not complements, but rather reconstructs the whole thing to finalize the process of DNA replication. Okay, so ligase is basically, not to personify it, but it's gonna assume the polymerase did everything correct and just basically come and attach and finalize the process. All right, so in a nutshell, you have this together. Helicase has come, split it apart. This side and this side are going to be the old strands of DNA, right? So that when polymerase comes and checks everything, it's going to make sure G fits with C. And look what you have on the other side, G and C, A, T, A, T. So, thymine, everything is basically where this side is what? Just like this side. By using this as your template and building these fragments on, okay, the two different strands, it's one strand, but the two different single strands can be combined together, and they're what? They're going to be identical. This one is going to end up being exactly like this one, as long as polymerase caught any errors and so forth, and that they were corrected like they should be. So, your DNA is your genetic material that we should know already, consisting of your 4 different nucleotides. You're probably going to see on the EOC just the letters A, C, T, and G, but it wouldn't hurt to know their names as well. Each nucleotide is going to consist of a sugar, phosphate group, and one of your 4 nitrogenous bases. Okay, either adenine, thymine, cytosine, or guanine. Again, A pairs up with T and C pairs up with G to form what we call these rungs of that twisted ladder. When this curls up and forms this right here, you have what we call our double helix. Before cell division occurs, remember I told you mitosis spends 90% of its part in interphase. It's because DNA needs to have done this just before mitosis can occur. So before cell division occurs, DNA is going to copy itself in this process called DNA replication. During replication, helicase forms the replication fork. The 2 strands are going to split apart, forming the rules of base pairs. A has to go with T, C has to go with G. These Okazaki fragments are going to come and build up, and then it will retwist back to this again. Okay? But when it retwists back to that, what? New genetic material has been added. Therefore, now, since it's in essence, since it's bigger, when cell division occurs, now there's an equal amount of genetic material to be distributed to the parent cell and the daughter cell. And like I showed you in that last picture, the two new strands are gonna what? They're gonna be identical to each other. So this is why in mitosis, that parent cell and that daughter cell will what? Will be identical to each other. Okay, and we basically answered this question right here. By these enzymes, making sure, building it, proofreading it, fixing whatever needs to be fixed, and then basically attaching and re-zipping this thing. Basically, since you have the old one as the template, you can't, it's not going to make a mistake. Each strand of DNA has the information needed to make that base pair. So because each strand is used to make the new strand, they're said to be complementary of each other. Okay? All right. This is going to be the end of DNA 1. At this point, then, look for the video of DNA 2, which is going to encompass transcription and possibly translation.